for asking questions is going to be complicated. So please do follow the guidance from Francesca. Ask your questions in the chat. That would be the ideal thing. Thank you.
So please mute yourself because we cannot hear the background noises. So except the speakers, please mute yourself. You can unmute yourself later on when you want to ask a question, but now everybody please stay mute. Okay. Thank you. Welcome to the speakers, and we will hear from Andrea Carfi first. Thank you.
Okay, thank you. Let me share my screen. Presentation mode. Just a second. I hope you can see my screen. Let me put it on.
Yes, we can.
Okay. So first of all, thank you for the invitation, both to Giovanni and Francesca. We've tried to, organize this meeting for some time, and it's great to be here today. So I'm not going to necessarily, say too much about the Moderna vaccine. I'm happy to talk about it during the question time. But what I would like to do is to just give a short background on the virus, infection, vaccination, and the differences, and the vaccines that are under development at this point with the most advanced, and what we can expect, what are the next steps and what we can expect happening in the next weeks and months. As a background, the coronavirus, and all of you have heard a lot about it, is a large virus, one of the largest infecting humans. Is a so-called envelope virus as an external envelope. Let me put also the pointer here. So as an external membrane that protects the genetic content of the virus. This is the RNA that encodes for the proteins of the virus. On the surface of the virus are the different viral proteins, but the most important is the spike protein, the so-called S protein, which allows the virus to bind to cells and to enter within two cells. The spike protein is the main target for antibodies, for neutralizing antibodies, so antibodies that can prevent infections, and is basically the main target for most of the vaccines under development at this point. So, of course, the coronavirus is a family of different, viruses. Generally the host is not humans, but they can jump from animal reservoir to humans. And this has happened already a couple of times in the past, first with the SARS, then MERS, and most recently with SARS-CoV-2. But there is, of course, a difference in number of cases between the SARS and the MERS, and now with the SARS-CoV-2, and mostly due to the fact that this virus, SARS-CoV-2, can be transmitted very easily. And so, at this time, there are more than 50 million people that have been infected with SARS-CoV-2, and I think more of 1 million people that have died because of this infection. So one of the questions, of course, in the field is if in the future there will be additional infections from other coronaviruses. And this is something that, of course, we all have to be prepared for. So the virus causes a form of pneumonia. And this is because the virus can infect basically the lung epithelial cells. And this is due to the fact that the spike protein can bind a receptor on the surface of the cells, called the AC2 receptor, and after binding can enter in cells, and this leads to inflammation and then also to pneumonia. But this virus is a really systemic infection, so it doesn't infect only the lungs, but also other organs, the nose, the gut, CNS, heart, and circulation. So leading to even other forms of disease, even myocardial infarction and inflammation of the endothelium. So different manifestations caused by the same virus. And so how the body reacts to the infection. So the virus, so this is a respiratory infection. The virus enters through either the respiratory tract, the nose, the mouth, and once the body gets in contact with the virus, this elicits an immunoresponse. So it's seen as something foreign, and therefore there is elicitation of antibodies, and this antibody can neutralize the virus, can basically clear the virus. In some cases, of course, the immune response is not sufficient, and therefore, you can have a high level of virus, and this can lead to hospitalization and some cases of death. But if you recover from the infection, then you will have antibodies in circulation, neutralizing antibodies, and you might be protected from a subsequent infection. The main response to the virus is basically through generation of antibodies that again can block the virus and prevent infection, but also so-called cellular responses that basically recognize fragments of the virus and infected cells, and therefore there are cells in the body that allow clearance of those cells. So you can also kill the infected cells. So this is the case of an infection. In the case of vaccination, we are doing something actually similar, but instead of giving to the body basically a virus, we give a piece of the virus, sometimes a protein or multiple proteins. And here it's simplified with the spike protein. Again, the spike protein is seen as a foreign, and this leads to an antibody response and hopefully a neutralizing response. So one of the differences between infection and vaccination is that in the case of the infection, you may have a damage at the level of the immune system. So there are antibodies that are being raised, but because there is a damage, then the antibody response might be weak or waning quickly. In the context of vaccination, you're just giving, again, you're not giving a virus, there is no infection going on, and therefore there is no damage of the immune system, and hopefully the responses are more persistent and more robust. So the immunization might give a better immune response than actually an infection. So one thing that I want to mention on top of the vaccination is also the concept of a passive immunization. In this case, what we do is isolate antibodies from a subject that has been infected by the virus, and these antibodies, of course, can be produced by recombinant technologies and given to a subject. So this is so-called passive immunization, and therefore these antibodies that now are in circulation may prevent infection from the virus. There are a couple of these antibodies that are under development, and most recently Eli Lilly got an emergency use authorization for one of these approaches. So now, how do we develop a vaccine? So we, as I mentioned, the concept is giving, of course, a piece, a fragment of the virus to a person to elicit an immune response. But to develop a vaccine, there are several steps that we have to go through. So first of all, there are animal studies that have to be performed. This is to demonstrate that the vaccine is safe, that it raises immune responses, and that is protective. And we do that in different animal species: mice, ferrets, hamsters, and monkeys. And some of these animal species are susceptible to the SARS-CoV-2 infection. So they can be infected with the virus, and they develop symptoms that are similar to what we have in humans. Then the vaccine has to go in a clinical study, in a phase 1 study, where the main goal is to show that the vaccine is safe, it's well tolerated, and we start getting the first immunogenicity data. So we verify that actually there is an immune response against the vaccine antigen. If the phase 1 is successful, we will move it to a phase 2. Again, we confirm safety, tolerability, but now it's more about immunogenicity and deciding how much of that vaccine has to be given to people to elicit a robust and persistent immune response. The next phase is a phase 3 study. which is an efficacy study. And these are much larger studies where we basically look at prevention of disease. So we want to demonstrate that actually the vaccine is preventing COVID-19 in this case. But at the same time, we're still looking at safety of the vaccine. And so as you will see, all through the development, safety remains one of the key parameters that has to be followed. The vaccine is given to healthy people, so as to be safe, and that's of course is the most important criteria of all. If the phase 3 works, and so you demonstrate efficacy, then there is the vaccine can be approved by regulatory agencies. Here in the US is the FDA, in Europe, EMEA, and so on and so forth. And then you can start using the vaccine actually. But what you have to also know is that studies don't finish when the vaccine is approved, but there is still a commitment by the companies that develop the vaccine post licensure, and so we have to still monitor safety and the protection coming from the vaccine, as the vaccine now is going to be used in a much larger population. So what about now the COVID-19 vaccine pipeline? There's a lot of vaccines that are being developed, as you all know. At this point, there are more than 59 that are in human trials at different stage of development, and another 40 that are in preclinical studies. So quite a bit of vaccines. But keep in mind that this vaccine is going to be for a very large population. It's the entire world, over 7 billion people. And therefore, we will need many different vaccines that can be used safely and that they are protective. So most of the vaccines are in early stage development, but there are some that have reached late stage development, and that means phase 3 studies. But at this point, none of the vaccines has been approved. So what are the technologies that have been used? I mean, and I will focus on the ones that are really being used in the context of the COVID-19 development. So there are vaccines that are basically based on the virus itself that has been, inactivated by, different methods. It might be chemical inactivation, and basically that's what's given to the subject. Sinovac, that is a Chinese company, is one of the companies that has been using this approach. This is a sort of an old-fashioned approach that has been very successful for many of the old vaccines. Then there are vaccines that use recombinant technologies, and in this case, what you are giving is just one protein, the spike protein, sometimes with adjuvants, molecules that boost the immune response. And Novavax and Sanofi are the ones that are most advanced with this approach. And then there are more technology that have more recent, and one is using inactivated viral vectors. This is the approach used by AstraZeneca. I know some of you are familiar with the site in Pomezia where some of the manufacturing for this vaccine is going on, and Janssen. In this case, what you are doing, you use a virus that is not harmful to humans, like a virus that causes common cold, that has been inactivated, but now it can express one of the proteins of the SARS-CoV-2 virus. So when this virus enters into cells, expresses one of the, in this case, the spike protein, and that elicits an immune response. And finally, we have the genetic vaccines, and this is, one of the most advanced is the technology of mRNA, and Moderna is, of course, using that, and BioNTech, Pfizer as well. And in this case, basically, there is a lipid nanoparticle that basically delivers to cells a piece of mRNA that expresses one of the proteins of the virus, again, the spike protein. So in this case, there are no proteins that are being delivered, and the technology doesn't really require any cell culture. So the vaccine can be basically produced in vitro without need of large scale cell culture. So the two approaches on the left, so the inactivated viral vectors and genetic vaccines, are actually at this time the most advanced, and because of this technology actually allowed to move very quickly. Because again, as I mentioned, you are more platform. You don't need to redevelop the vaccine every time. And in the case of mRNA, you don't need to generate, you don't need a large manufacturing site. You don't need. Let me just focus on the the vaccines that are most advanced: the Moderna, BioNTech, AstraZeneca, and Johnson & Johnson. So 2 mRNA vaccines and 2 viral vectors. All these vaccines are in late stage clinical trials in phase 3, both Moderna and BioNTech, and also J&J and AstraZeneca. And these trials really include a large number of subjects: 30,000 for Moderna, 45,000 for Pfizer, and more than 60,000 for the viral vector vaccines. The vaccine studies, I mean, for phase 3 have started all, during the summer, between July and September. And so we are basically now at the point where we're going to get efficacy data for all these vaccines, and we're going to see how they work. And as you may have heard yesterday, Pfizer has announced some initial data, interim data, showing that the Pfizer mRNA vaccine is giving 90% efficacy in prevention of disease, of COVID-19 disease. We will see soon the data from, of course, hopefully from Moderna and from the other technologies. But I would like to point out that, while the vaccines were being developed and, moving through clinical stages, all these companies have started manufacturing at risk. And therefore, meaning when I mean at risk is that manufacturing without knowing that the vaccine was going to work. And I'll mention how that is possible. But we expected that several million doses of vaccines will be available by the end of the year. Pfizer has committed for 50 million doses by end of the year, Moderna 20 millions, and several millions also by other manufacturers. Going to 2021, then the US government has also bought a large number of vaccines from this company in the hundreds of millions of doses. And the same is happening in Europe. So in the 2021, through 2021, there will be billions of doses that will become available. So I would like to just mention, to finish, how this was possible. I mean, I think, as you all know, the development has been very fast, and compared to other vaccines and vaccines in the past. And this is not because of we have taken shortcuts in terms of safety, but just because there was a large investment. And this has been in part possible because of the Operation Warp Speed, supported by the US government, which has as main goal to deliver 300 million doses by January 2021. And this has allowed to, as I mentioned, to do simultaneously, clinical development, but also manufacturing. And there has been more than 10 billion investment in the vaccine and antibodies to make it happen. And the operational warp speed that is led by Moncef Slaoui and Gustav Permer, General Gustav Permer, is really supporting different aspects from diagnostics, therapeutics, and vaccines, in collaboration, of course, with NIH, BARDA, and the CDC for the delivery of the and administration of the vaccine. So next steps will quickly, so we'll have hopefully efficacy data. Once the company has the efficacy data, they can apply for an emergency use authorization. So they will ask to deploy the vaccine before the vaccine is fully approved. Is a temporary approval, if you wish, in consideration of the risk benefit of the vaccine. After that, after some time, if a vaccine company can apply actually for licensure, a so-called BLA, and that is when the vaccine basically becomes licensed and becomes a product. All the trials so far have been in adults, 18 to 71 plus. But some companies have gone down to 12 years of age. But of course, one of the next steps will be to go to children's, and also immunocompromised populations, also pregnant women, and so on and so forth. But that will be the next step in the development of these vaccines. And then, of course, there is distribution that will be coordinated by CDC and Operation Warp Speed. So I'll stop there. I hope I gave you a sense of where we are with this. And I'll be happy to take questions maybe after Alessandro has presented. Thank you.
Great. Thank you, Andrea, for this very clear presentation. I think you really explained very simply to all of us, also for people who are not in the science field. So I have a lot of questions already in the chat, so we can go through some of them maybe before the second presentation, because these questions are mainly related to what you talk about the vaccine. So there is one question about are vaccines likely to fail during clinical trials as drugs? Same as drugs?
I'm not sure. So if vaccines in general, let me rephrase the question if I understand. So if vaccines in general have chances to fail, or the coronavirus vaccines? I think, as any drugs, you don't know until you have done your efficacy trial if the vaccine is going to work. Of course, animal studies might de-risk when there are the right models. You don't have animal models for all the vaccines that you're trying to develop. Having those protection data, for instance, in the case of the COVID-19, clearly gave a lot of hope in the human studies, but you don't know until you do the trial. I mean, in the case, as an example, in the case of the COVID-19 vaccines, what FDA gave us threshold was 50% efficacy. And we would have all been happy with 70% efficacy. The fact that we see 90%, that tells you about the fact that it's a little bit unpredictable, and also how these things can change. But there are vaccines that have failed in clinical studies, HIV being one of the most.
Okay. Another question is the phase 3 clinical trial also examining the need for multiple vaccinations? So is it likely we will need more than one shot, or you think, at least, I guess, the Moderna vaccine, it will be one shot, or you need multiple shots?
So there are two questions there, right? So most of the vaccines that are being developed are 1 month apart. So you need 2 immunization, and that's the protocol, the approach that is using in the phase 3 study. There is for the adenovirus approach, the phase 3 study is with a single dose, so a single injection. J&J is also trying 2 doses in a different phase 3 study. So that's the first question, right? How many injections you will need? So for most of the vaccines, it's going to be 2 doses, 2 injections. Then there is the question about the persistence of the response. So, once you receive the vaccine, for how long are you going to be protected? And that's something that we don't know at this stage, right? We will have to look at the immunoresponses during, with time, and see if the antibody and neutralizing antibodies remain there, or if they decay. Generally, this is something that you assess, with time. And, in some cases, for some vaccines, you need a so-called boost. So, after a number of years, maybe 5 years or 10 years, you need a new injection. As you know, in the context of flu, you need annual vaccination. So for SARS-CoV-2, we all hope that it's going to be multi-years, several years before it boosts. But we will have to find out in the real life, and once the vaccine is deployed.
There are a few questions that I try to summarize together about, of course, safety, which kind, if any, anticipated side effects that you would think about, that you could anticipate or you would expect, or long-term side effects, and also what's the difference really between, since the Moderna vaccine is slightly different than the classical vaccine development with the whole virus or with the spike protein rather than nucleic acid, do you expect any other side effect or any other compared to?
As I mentioned, safety is the most important aspect, one of the most important aspects, if not the most important aspect of any vaccine. So there is I would say that is the aspect where there is more scrutiny, and clearly the safety is what regulators and users are going to look at. In the phase 3 studies so far, and in the Moderna study, we haven't had any serious adverse event. Most of the side effects are what you normally see for other vaccines. Maybe some pain. You might have, in some cases, fever, myalgia, but nothing that is long-lasting. Generally, the symptoms go away within 24, 48 hours. So far, the vaccine has been very safe. Of course, now the vaccine has been tested in more than 30,000 subjects, so we have a much larger safety database. One of the requirements, even in the phase 3 study, is to follow people for other 2 years. So we will follow also safety, not only in the short term, but also in the long term. The last thing I want to add is that for the mRNA technology, first of all, we have now 30,000 subjects from the Moderna, 30,000 from Pfizer. There are many other mRNA vaccines that have been developed, but also, before even starting the work on the SARS-CoV-2, Moderna had more than 7 vaccines in clinical trials, and we had safety data for more than 3,000 subjects. So, and immunogenicity data. So I think the data so far have shown that it's a very safe. I could go on the safety, of course, but I think one aspect that I like is that we don't use, cells. We don't use virus. The vaccine is basically, produced with processes that are well-controlled and well-controlled reagents. So, in my opinion, is actually safer than many others.
But did Moderna ever produce a vaccine with this technology before?
Of course, Moderna has generated, as I mentioned, vaccines for other infectious disease, but we did not have, and we don't have a licensed product. So this will be the first licensed product.
That's great.
But that's, of course, has been, and I would say also for Pfizer, has been actually one of the achievement and the challenges to, scale up the technology to be able to make, hundreds of millions of doses. And, really kudos to our manufacturing team that has done that, internally and in collaboration with with partners at this point.
Yes, so that brings me to other set of questions which regards the distribution. So several people are asking how they will be distributed. Will they be? Is gonna be based on, of course, who is gonna be approved first, who's gonna get it first, which countries are gonna get it first, has to do with FDA versus European agency approval, and the process of approval is gonna be the same, it's kind of unified, or?
Yeah, it's a lot of questions there. So let me try to, so I mentioned the process. I mean, I think, the first thing is to get efficacy, and then you have to, present all the data, the efficacy data, the safety, the phase 1 data, phase 2, phase 3, and, go to the regulatory agency and get, in this case, what would be an emergency use authorization, which means that you can start using the vaccine, and not necessarily for the general population, but just because of the risk. I mean, the current pandemic is at such a risk, for the population that is worth implementing vaccination. So that's the first step. That's a decision that the regulatory agency have to take, and they're going to take it for each vaccine separately. Then once the vaccine, is approved for emergency, then there is, of course, in terms of deployment, at least in the US, is going to be through the, in the pandemic situation with the CDC and Operation Warp Speed. I think there is going to be, there's going to be, it's not going to be used in the general population immediately. There's going to be, criteria and who should receive it first. I would think, healthcare providers in the front line and probably also the elderly are going to probably be the first ones because are the most, the population at risk. In terms of, I mentioned that I think, by the end of the year, there will be, millions of doses available, but many more in 2021. So I think starting in 2021, there will be the opportunity to vaccinate, a larger part of the population if the vaccines have been, of course, approved, or those that have been approved. In general, I think, most of these vaccines that I talked about, have been developed in the US, but there are also, agreements with the European community. So I would expect that vaccination, as far as the vaccines again are approved by both regulatory agencies, will be used, almost at the same time in the US and in Europe. The timelines, I think, we are in November, there is first the emergency use authorization and then deployment. So, best case scenario, I think, will be towards the end of the year for the first vaccination, and then many more in 2021.
Are you actually testing the vaccine in different populations?
Yeah, I think thank you for the question. That's an important actually question. So I mentioned that in the phase 3 study, I'll talk more specifically about the Moderna study, but that's true also for others. We have 18 from above 18 years of age, and includes elderly. But the way the study is designed is that first of all, more than 40% of the subjects enrolled in the study have to be at risk, either because they are above 65 years of age, or because they have a comorbidity that put them at risk. So that was one of the criteria. And then, and we actually slowed down the trial to make sure that we accomplished that. The other part is diversity. Of course, the trial has to be representative of, the US population in terms of, minorities, Latinos, Black American. And even there, I think we have been able to achieve a distribution of the population that is representative of the United States. And if people are interested in our in the Moderna webpage, we have really the distribution that we have achieved and the progression of the clinical study. Similar criteria are going to be used by other manufacturers as well.
And are kids included? They can be vaccinated too?
So the Pfizer vaccine includes down to 12 years of age. So they expanded the trial to also include the 12 years old. And but if you want to go, to a lower age, that will be additional clinical studies. And so you have to demonstrate, safety and tolerability in that population, which might respond differently to the vaccine. And therefore you might need to test a different actually dose level. And generally, because of course the children, you want to be, even more careful. There is a age escalation, so you don't go directly to, of course, to younger kids, but you go gradually, and by monitoring immunogenicity and safety, and then you go came down, and so on and so forth. So it will take a few more months before vaccines will be tested and at some point approved by regulatory agencies. There's also a question about what is needed, what data will be needed to have approval of the vaccine in children. If immunogenicity is sufficient, or if you need efficacy, and so on.
And this RNA-based vaccine can be given to people with autoimmune diseases. Somebody is asking.
People with autoimmune diseases. I mean, I would say that it shouldn't be different than any other vaccine. I mean, yes, I think you should be able to give it to immunocompromised and people with autoimmune disease, but has to be tested. I'm not sure this was included in the current trial.
Okay, great. So we have so many questions. We will be here for 3 hours, so we have to move on. And maybe we can come to some of the questions at the end of the second presentation from Alessandro Vespignani. So Dr. Vespignani is an expert on the modeling of the pandemic. So he can maybe explain to us how the pandemic will be affected by vaccines, or in the next few months, how it's going. Thank you.
Good. Thank you very much. I will share my screen now. It's very, let me open with one, I think you should be able to see the...
Yeah, we can see.
Yeah, so great. Yeah, let me say, if you, and I think Andrea will agree, if you think how the vaccine will affect the pandemic in the next few months, is probably the vaccine will not affect the pandemic in the next few months. And that's because there is no time for a massive deployment in the world population before before the next few months of the 2021, and actually even for the first couple of months, 2-3 months of the 2021, I don't expect you please do not expect to be in line waiting for your vaccine. As Andrea was mentioning, there will be a hierarchy of distribution that will start with the frontline workers and then go down the list. So unfortunately, this is perhaps a little bit the bad news with respect to all the good news that we got from the vaccine side, is that essentially for the winter, we still have to cope with the pandemic the old way. The same is a little bit for Andrea mentioned the monoclonal therapies, etc. Again, they will be used, but let's not assume that we will be flooded with hundreds of thousands of doses of those therapies. So we still have some work to do, in which we have to mitigate the pandemic in different ways, and especially with our behavior. Now, let me first of all start saying that what I will show is a very large scale collaborations that we developed along the years, and now in the last probably in the last 1 year, mostly working 99% on COVID. There are many groups involved in the US, across the world, and let me say that there are several Italians, just since we are talking about in the PIB context, several Italians involved. Some of them are in Italian institutions like the FBK Foundation, the ISI Foundation, and some here in Boston. For instance, Matteo Chinazzi is working on Northeastern, is one of the person that is really pushing the envelope on the data and modeling side. So I'm in a sense, I'm proud of the Italian representation of in the work here. So what we do, let me try to give you in two sense what is our work, because our work is actually to take data, models, and transform those data and models in possible intelligence against the disease. In a sense, what we are trying to do is to take a timeline of data that could be reporting of deaths, cases, whatever, and transform into something that is more evolved, like weather forecast, where you don't have just you know, the value of the epidemic in the next couple of days, but also how to say the trajectory in terms of space and location, what we call the where, when, and how much of a pandemic. Well, to do that, we use models, and those models ingest data that are coming from a variety of sources. So here is in a nutshell, in a visual nutshell, what we do. We describe the pandemic evolution at the worldwide scale. We integrate data that comes from very highly detailed population maps. We have data, real-time data about airline, domestic and international flights, commuting patterns. We use mobility data that comes from different location intelligence providers. And all that recreates in the computer a kind of synthetic world where individuals are really going around, and since we are the carrier of the disease, you know, we can track and monitor how the disease spread, and then how the disease unfortunately activity progress in different areas of the world. The model works at very different scales, so you can work in certain cases at the global scale, and that was most of the work that we did initially to understand the potential, the pandemic potential of SARS-CoV-2, and then, you know, you get to zoom in specific countries or even urban areas in order to address a specific question for the policymakers. At the basis of all that, it's really important to stress that there are data. And so there is a lot of data that comes from the census data. What we use is micro census data. So it's really a kind of tomography of the society in different places of the world, where you really have all the information about thousands or hundreds of thousands of households. You have census data like the age distribution, the frequency of the size of households, etc., etc. Out of those data, what we construct is what we call the synthetic population. That is a map of individuals and how they are connected and how they interact. And here it's very important to consider also data that provides proximity, mobility in terms of all our daily commuting patterns, or the way you work, the work structure, the school structure, and so on and so forth. You can decide to work at this very microscopic level, and in some cases we work down to that level when we do model for specific metropolitan areas. And then otherwise you can work on aggregation that are a little bit more coarse-grained, as we say. So you average and you create what we call mesoscopic description. For instance, maps of depending on your age, what is the number of contact with other people of different ages. That provides the model the flexibility to understand what different impact the pandemic has across different age bracket. And this is one of the crucial points, as we know that the population that is most at risk is indeed unfortunately is in the elderly, as well as then some other strata of the population that have comorbidities and pre-existing condition. all that, all those data are integrated in a model of the infection transmission. The infection transmission model comes from the clinical data, where you have to integrate data concerning the incubation time, the probability that pre-symptomatic individuals will transmit the disease, the fraction of asymptomatic individuals, and so on and so forth. So all that is generally a large computational, is reflected in a large computational model that is used to make projections. And here it's, in a sense, again, in another nutshell, how does it work? Because, models are always models. They don't have to be confused with the reality. Models have always assumptions inside, and there is a level of uncertainty. The uncertainty is because of the initial conditions. We never know the exact number of infections at any point in time in a specific region. So we have always uncertainty to consider in the model, and we have also what we call parameter uncertainties. What are the parameter uncertainties? Yeah, if you think about what we knew about SARS-CoV-2 at the very beginning, we didn't even know the incubation time or what we call the serial interval, so the number of days between one infection and the secondary infection, and so on and so forth. So we had a lot of uncertainties initially. They shrink and they become, we know better the values of those parameters, but we never have a full exact knowledge of those quantities. And so these are a lot of uncertainties. And then there is another source of what we call stochasticity, so the randomness in the evolution of a disease, is the fact that you might have a reservation on a flight and you might miss that flight, or there is certain people that commute or might decide to stay home, and so there is also a level of uncertainty that is natural in the transmission of the disease, stochastic event. In some cases, you might get in one place and infect somebody else, and you repeat the same story, and you might have a different outcome because the infection, it depends on many infection process among individuals might depend on many things that you are not able to describe explicitly. So this stochasticity implies that we have to basically, like we do for weather forecast, to start with certain initial condition parameters and modeling assumptions, and then repeat in the computer those stories thousands or up to millions of times, and the evolution of those stories will provide us what is a map of possible futures, and then we integrate those maps to create a probabilistic projection of what could be the future. And in those probabilistic projection is basically embedded this uncertainty cone of what could be the evolution of the pandemic. That is extremely important because this is like for weather forecast. On your phone, you will never see tomorrow is going to rain. You will see that there is a 20% chance of rains or a 40% chance of snow. This is the same for pandemic. When you say numbers, when you say what will happen, is always percentage. It's always probabilistic future. And that means that you don't have an explicit deterministic outcome, but you have possible map of what will happen. Well, when we do forecast, is the things that I always tell everybody. If I would tell you that on December the 28th we will have 20 inches of snow in Somerville or Cambridge, you would look at me and you would say you are completely crazy and you are a crook, because you know that it's impossible to do that. Yes, and the same is for epidemic forecast. So, when epidemic forecasts have a time horizon that is about 2 to 4 weeks. And so you can project the number of deaths, number of cases on a time scale that is between 2 to 4 weeks. Why? Because it's extremely important in those projections to consider what kind of situation you have on the ground and what kind of policies are implemented, what is the behavior of people, etc., etc. Why you can assume that on a time scale of 2 to 4 weeks to be more or less under control, because you know that there will be implemented certain policies, or there are in place certain policies, or you measure the behavior of people through different data sources. When you start to project on a longer time scale, obviously you are making assumptions, and these are not anymore forecasts. It's a different things. So the forecasts are on 2-4 weeks, and this you see example of what the work that we do every week for the center, with the Center for Disease Control. State by state, you make projections, you have a confidence interval about what you will expect in terms of deaths and cases, and then you aggregate all those things to get something is at the United States level. This is, you see, these are projections that were, it's not an updated projections, although we publish every week projections on the Center for Disease Control website. This was at the beginning of the, unfortunately, of the, where we started to see an increasing trend in starting mid-October, and this is what we see more and more more increasingly so in these days. Okay, forecast, another good thing, so that you can understand how the work is done, is that there is no way to get only one model into the forecast. Like for the weather forecast, you know, there are always several models behind those. You have different groups simulating with different techniques, modeling assumptions, data simulations, the pandemic. And then you create what we call ensemble forecasts. So you take several of those forecasts and you compound together, and the hope is that they will point in a sense in the same directions, that they are consistent, and this is extremely important. And so this is like when you see, when there are the hurricane forecasts and you see those spaghetti models for the hurricanes or the touchdown in Florida or the Gulf of Mexico, this is the same. So you have different spaghetti that represent different models that assume different uncertainties and different assumptions on the disease, and then you see that most of them have have a coherent behavior, and on the basis of that coherent behavior you are more safely forecasting what will happen in the next 2 to 4 weeks. Well, these are forecasts, and actually, unfortunately, the United States is the only place where there is a operational, at the moment, effort of this kind. There is no in European, not even in European countries, and I think this is something that we are all advocating a lot to happen. So to have more knowledge of, at least on the what we call the short term. Then there is the scenario analysis. Scenario analysis is a different business. So it means that you use the model to study different situations in which you assume in the long range, in the long-term future, different policies, different behavioral changes of the population, and this is a different things, because this is not a forecast. This is creating a portfolio of possible scenarios, of possible evolutions that goes from generally, well, generally, actually, we are forced in crisis units and task forces to do what is the worst case scenario, in which basically you assume that the epidemic will spread unmitigated, to the most optimistic ones, in which you assume that all your interventions are layered one on top of each other and they work seamlessly. So that generally doesn't happen, and you have all intermediate ranges. So this is what is produced and provided to task forces across the world, so that then the policymakers can make a decision about what to implement. The final decision, the end decision point, is not just based on what is the best scenario, because it's also constrained by resources, it's also constrained by public acceptance, is also constrained by the economics, by resources, by the politics of the moment. You can imagine. So, and this is something that I really care to say, because in many cases you have probably heard recently, oh, we just follow the science. No policymakers is just following the science. There is always something that is much more complex. There are not just epidemiologists in those task forces, but there are also economists, there are engineers, there are logistic experts, etc., etc. And each how to say, policymaker has to make very tough decision based on those possible map of the future and what they believe it can be achieved or possible to achieve. And so this is the work that is a complex work that is done constantly. And it was done especially at the very beginning of the pandemic, where most of the states decided and governments decided to go for the what we call the most precautionary way. That was the one of the very heavy shutdown and lockdowns of a large part of the societies. And now this is with the approach of the winter, the idea is to try not to have those kind of very drastic measures, but actually by the more knowledge that we have of the system, of possibly with the preparation that happened during the past months of in different places of the world, to have different response to the epidemic. Unfortunately, this is not possible in a few places. Well, and here with the scenarios is where I can just connect to the previous talk about vaccination. And here it's very, again, something that I always try to tell everybody, you know, each one has to do his own job. We as epidemiologists are not in the development of vaccine. And so for us, basically, we just get at a certain point the fact that there are a certain number of doses of vaccine. There is a certain measure efficacy and effectiveness of the vaccine. And the point is to start at that moment to project what is the best way to distribute across society in each state or country, but as well across the world. Because let's not forget that we always think in terms of, in a kind of local perspective. So, you know, we have a vaccine manufacturer, but actually the vaccine manufacturer produces vaccine for the entire world. And so at this stage, the point is, in the case of a pandemic that is a global event, what is the best way to minimize deaths and minimize economic disruption? And it's not a given that you know, the distributing vaccine doses in a way that is just, okay, I buy a certain amount of doses and I try to amass doses in specific countries is the best way. So what we are going to do with modeling is to try to simulate those different scenarios. And we did work with the Gates Foundation and now also extending this work with other partners in order to see, well, what happens according to different allocation strategies. And one allocation strategy is, okay, who has more money, buy more doses of the vaccine. And so basically, you know, one association is high-income countries will get first most of the doses of the vaccine, and is one option. The other one instead is to say, well, you know, let's imagine instead that we can have a more equitable distribution. This is work that has been put forward by the WHO and by other groups, in which we have that the vaccine is distributed in ways that are, for instance, proportional to the population or proportional to other indicators of the course of the epidemic. Well, you know, if you do those scenarios, you see that there are very big differences in terms of, you have here, for instance, what would be a scenarios in which, in a sense, in a way, in the uncooperative, what you see here scenario, basically all the first 2 billion doses of the vaccine are distributed among high-income countries. And instead, you know, a cooperative vaccination scenario that mostly distribute vaccine across the world in a way that is more or less proportional to the population. Well, you see that, you know, with the more equitable approach, you have 60% of the deaths are avoided, while with the how to say, uncooperative approach, you get only 30% of the deaths averted. And that means that basically, you know, a cooperative scenarios has the power to avert twice as much death as an uncooperative one. There are many other considerations from an ethical standpoint that has to be done. This is just a very simple things that we did, and now we are looking at more how to say, detailed indicators of each in each country. It's important to say that here in these scenarios, what we did is exactly to go, as Andrea was saying, so that we distribute the vaccine in a way that is proportional to the risk groups. So when we sent a certain number of doses to a country, those doses are distributed accordingly to this hierarchy that goes from frontline workers to the high-risk groups in terms of age and so on and so forth. So what you will have is that the very large chunk of the initial stockpile of doses available are given to people who have the highest risk of complications and deaths and death outcome from the disease. There will be a lot of discussion how to do for the rest of population. And this is something that we are advocating with each country to start to work as soon as possible. So to create very clear timeline for the vaccine distribution. Well, these models consider a lot of other things like problems in the supply chain, distribution, that we know that there are a lot of things related to the cold chain for some of those vaccines. So there are many other complications that have to be taken into account, and that can be, how to say that these are major logistic problems that we will have to face in the next months that how to say are going out of our expertise as well as from the expertise of Andrea. I think, you know, you at certain point one ends out the stockpile of vaccine, and then there is the problem of how to distribute and ship across the world in places at a certain point that, you know, we don't have just to think about high-income countries, but other places where we have much more difficulties. Well, let me close with one way to say, you know, and this is very important, whatever we do with the modeling, it's something that, you know, models aren't to be considered as unquestioned oracles where, okay, oh, the model is telling me that, you know, they are models, and especially when we do projection, do not communicate a precise future, but, you know, a range of possibilities. And so always keep in mind that what the model is saying is not an unavoidable future, but actually maybe a map of the future. And in general, especially for pandemic and epidemics, we have a lot of possibilities to change the trajectory of those for the good. So even now, when you hear on television, well, there are models that project 300, 400, 500,000 deaths in the next few months. Always the first question that you should ask for is, okay, what assumptions are you making? Why those models are projecting those, and what we can do to make those projections not be the future, because there is always that possibility. So I think I can stop here. I did 20 minutes, and perhaps is even too much. So thank you very much, and I'm very happy to take your question.
Thank you so much for the overview of very complex concepts, but you explained so clearly. So there are a few questions. I try again to summarize together. So there are a couple of people asking about the use of artificial intelligence, maybe in predicting risk, and how the use of you know, risk prediction algorithm to, you know, to the analysis that you do, and if it would be better to use this kind of technology maybe to identify hotspots, so that the vaccine will be distributed based on hotspots rather than population at risks. And the second part, if this kind of your scientific approach, your modeling can also be used to create an effective tracing system.
Well, that's a lot of questions. So first of all, use of artificial intelligence. There is a lot of artificial intelligence in what we do. The artificial intelligence is used also to generate data. So for instance, the high resolution maps that we use that now goes down to the level of a few miles times a few miles, are maps that are generated not just by census data, but by artificial intelligence algorithm that compound the census data with the light emission captured by satellites and with many other information. So, there is a lot of artificial intelligence in the process. Then there is a lot of artificial intelligence in the predictive algorithms. So, for instance, in the CDC, the Center for Disease Control Forecast Hub, several of the methodologies are artificial intelligence approach. Some of them are more black box than others. Some are more open. With artificial intelligence, there is a problem. If you have algorithms that are too much of a black box, you have to be very careful, because here we are working with public health, and we want to have be prescriptive on policy making and other things on the base of things that we can interpret, not just something like an outcome. But there is a lot of work now in artificial intelligence where there is a kind of combination of approaches, so that basically you can open the black box and get understanding. For instance, we have two approaches. The one is fully mechanistic, as we say. It's along the lines of what I said and show to you. So we simulate all the individuals, but then we have another approach in which team up with the University of California, San Diego, in which they take our predictions, the mechanistic one, but then they feed those predictions to a neural network that reads both the prediction and the actual outcome of the historical data. And in a way, it's able to correct our prediction for possible biases and for the fact that, there are biases actually both in the reporting of the real data and both in the modeling that we have. And so you can use artificial intelligence to optimize your approaches. Another thing is to use artificial intelligence as a way, for instance, to capture parameters of models, to create a more detailed ground truth with respect to what we have. Don't forget that we get data which are very delayed. The deaths that we are seeing now are deaths that have occurred weeks ago in some cases. In some states, there are up to 3 weeks delay in data reporting. And so, machine intelligence, artificial intelligence is really helping a lot also to cure those and have much better inference on the data that we work with. And so there is a lot of approaches. Yes, models helps also for the structuring of contact tracing and to testing strategies. We did a lot of work in the past, and many other teams have done a lot of work in the past months. For instance, we developed a model for the Boston metropolitan area that is based on real data from users that where we can really look at the census block level, where people are going, where they are in proximity. And that way you can do and optimize, you can provide what kind of level of contact tracing it would be necessary to keep the epidemic in a corner while at the same time you reopen society. And you provide estimates of the number of tracers that you would need, the level of testing, and so on and so forth. And that has been done in many places. Unfortunately, I have to be honest. Well, in Massachusetts, we have a good test and trace program, but in many places in the world, unfortunately, I think that the test and trace programs were not how to say, conceived to be adequate to what we were expecting for the winter. And actually, if you look at the epidemic trajectory now in Europe, unfortunately, including Italy, the test and trace has broke down, and at that point you have problems, and then we go back to large population mitigation measures as we know.
But in your models take into the account that if people don't want to be vaccinated, so vaccine hesitancy, they are asking. So I guess that's part of the behavioral part or of the assumptions, like wearing mask, taking the dose when you are supposed to take it or not. That's
yes. The model try to take into account this by using effective transmissibility because of the widespread use of masks, the fact that the vaccine might not be administered at the pace that you have planned. These are all things you can do, and actually this is part of the value of models. The value of models, many people think that models are used mostly to get the forecast, but actually in public health, the use of the models is mostly to look at, okay, and if I have vaccine hesitancy for 50% of the population, what would happen? And these are the questions that you ask the models, and the models are the only one that can provide some answer, at least some guidance, some evidence-based elements for the reasoning. Obviously, those parameters in some cases are really parameters for which you do what we call a sensitivity analysis. So we look at different scenarios in which the hesitancy is very high or medium, very low, and then we see at the different outcome and the different impact of vaccination campaign on the epidemic, and so on and so forth. So that's part of the work that modeling groups across the world are doing at the moment.
Great, thank you. So since we are way above the hour, we need to conclude. So to conclude, just one last question. If we assume everybody is taking the vaccine is successful and is effective, and it will be available for everybody in March, can we go back to normal life using masks?
Oh, if we would have billions of doses in March, and then you have to ask Andrea if that's possible, we would be in a different world. I think we will not have billions of doses, but we will start to have an important stockpile of those. No, let's hope for that. I think we have to be fairly optimistic for starting in the spring. We have seen that there is a very seasonal behavior for the virus, so we can use those months to to roll out the vaccine and to prepare. But I think for the Northern Hemisphere, we have always to think globally. Northern Hemisphere is one thing, the Southern Hemisphere is another one. When we will we will transition to the summer, they will transition to the winter. So we need to see. I think that now we have to focus really on fighting this for the next 3 months. That will be winter, and we need to have a lot of collective responsibility. We need really to think about collective behavior, masks wearing, avoid crowded places, no indoor places, and be patient. It will still, we still have a couple of few months ahead that will be a rough ride, but then I'm thanks to people like Andrea, we will likely have the weapon to to send back this virus to history, hopefully.
Yes. Okay, and with this positive note, a note of hope, and we really would like to thank the speakers. Thanks both of you for participating tonight, but for your work, it's really amazing to see Italians involved, and you know, you make us proud, all of us, and you know, you do such important work in this time.
Thank you.
Maybe before we close, first of all, thank you again, Alessandro and Andrea, for making time on your busy schedule. Before we close, we want to, you know, with Professionisti Italiani at Boston, we have organized up to 4 online events per week since the pandemic started, to help all the community, English and Italian, to have things to do during the lockdown and feel together. We are planning a very interesting seminar before Thanksgiving with executive chef Marisa Iocco. She's co-owner of the restaurant Spiga out of Boston, and she will present either next week or the weekend before Thanksgiving about how to make your Thanksgiving dinner in Italian.
Wonderful.
And she will show us how to actually make that happen. So stay tuned. We'll also have a follow-up event on COVID with another Harvard professor. I'm not going to say the name until we have it, the name on a flyer, but she's going to talk in January about how governmental public policies can influence all that we're talking about. And this is not about politics, of course, it's about enhancing public measures. So stay tuned. With PIB, we'll send information about the next events. Thanks again. And Francesca, maybe thanks again to the Comites and Professionisti Italiani of Philadelphia for working with us today to organize this very interesting event. Thank you.
Organizers, and I also want to thank the PIB managers that are behind the scenes here, you know, Federico, Chiara, Sonia, everybody, Nicole.
Alessandro, who's in Italy and connected from there to help organizing the event.
Exactly. And if you want to listen again this presentation, because maybe you missed something, we are going to upload a recording on our website. So go on piboston.org. And if you are not a member PIB, you can also join. It's free, and you will receive our newsletters with our events. So thanks again, everyone. Thanks to the speakers. It's really been an honor.
Thank you very much for inviting us. It has been a real pleasure to be among you. Thank you.
Thank you again. Thank you for the invitation.
Everyone. Bye. Bye-bye.
Bye.
Bye, thanks.
Bye.
Bye.
Bye-bye.